Welcome everyone to your own channel, ADP for Learning. I am your own learning partner cum host, Ashish. So, let's begin our learning journey. Today, we will learn 4 different terms that are associated with genomics and genetics. Starting with the first term, that is base pair. A base pair consists of 2 complementary DNA nucleotide bases that pair together to form a rung of the DNA ladder. DNA is made of 2 linked strands that wind around each other to resemble a twisted ladder, a shape known as a double helix. Each strand has a backbone made of alternating sugar, that is deoxyribose, and phosphate groups. Attached to each sugar is one of 4 bases: adenine, that is A, cytosine, that is C, guanine, that is G, or thymine, that is T. The 2 strands are held together by hydrogen bonds between pairs of bases. Adenine pairs with thymine, and cytosine pairs with guanine. One copy of the human genome consists of approximately 3 billion base pairs of DNA, which are distributed across 23 chromosomes. Human chromosomes range in size from about 50 million to 300 million base pairs. Because the bases exist as pairs, and the identity of one of the bases in the pair determines the other member of the pair, scientists do not have to report both bases of the pair, which is why DNA sequence is typically represented as single strings of letters. DNA sequencing involves determining the exact order of the base pairs across a DNA segment of interest, or across an entire genome. A signature goal of the Human Genome Project was to generate the first high-quality sequence of the human. The effort was successful in generating such a sequence for over 90% of the human genome, but it took nearly 2 more decades to sequence the remaining bits of the human genome, which were heavily enriched for highly repetitive and difficult-to-sequence stretches of DNA. The second most popular term is bioinformatics. Bioinformatics, as related to genetics and genomics, is a scientific subdiscipline that involves using computer technology to collect, store, analyze, and disseminate biological data and information, such as DNA and amino acid sequences or annotations about those sequences. Scientists and clinicians use databases that organize and index such biological information to increase our understanding of health and disease and, in certain cases, as part of medical care. The role of bioinformatics in biological research can be compared with the role of data analysis in the age of information and the Internet. In earlier days, the primary challenge was getting to the information. Advances in reading DNA sequences have lowered that barrier substantially. Going forward, the challenge is how to understand and interpret the information that has been collected. Because the data sets are large, whether you're talking about information about website visits or the human genome, computer-based methods are the default approach. In the end, bioinformatics work with human genomes seeks to discover practical insights about human health and biology with all its complexity. The third term of our today's glossary is birth defect. A birth defect, also called a congenital defect, is a physical or physiological abnormality present in a baby at birth. Birth defects can be caused by genetic factors, prenatal events during pregnancy, or a combination of both. Some birth defects are easy to see, such as an extra or missing finger, while others, such as an enzyme deficiency, are identified through special tests. Also known as congenital disability or congenital condition. Congenital conditions sometimes include some kind of impairment, but many do not. Congenital conditions simply refer to structural changes which affect any part of the body, such as the hand, leg, or foot, or which influence the development of some aspect of physiology, or how the body works. Congenital conditions, regardless of cause, are relatively common and can typically be identified at birth or shortly afterwards. These conditions are also quite treatable in many instances. And individuals who have them, especially under the care of a physician, often experience typical growth and development. The last term which we will cover today is BRCA1/BRCA2. BRCA1 and BRCA2 are the first 2 genes found to be associated with inherited forms of breast cancer and ovarian cancer. People with mutations in either BRCA1 or BRCA2 have a much higher risk for developing breast, ovarian, or other types of cancer than those without mutations in the genes. Both BRCA1 and BRCA2 normally act as tumor suppressors, meaning they help to regulate cell division. Most people have 2 active copies of these genes. When one of the 2 copies becomes inactive due to an inherited mutation, a person's cells are left with only 1 copy. If this remaining copy also becomes inactivated, then uncontrolled cell growth results, which leads to breast, ovarian, or other types of cancer. Everyone actually has a BRCA1 and 2 gene, and in their unmutated form, they carry out very important functions related to keeping your genome healthy and intact. Thank you for joining with me for the amazing journey of learning. In the future, we will again meet with other new, innovative topics. Meanwhile, don't forget to like, share, subscribe, and comment.